In this video we will discuss about mRNA translation process, and we will also discuss uh, how this mRNA translation process gets affected. So let us start with the normal global protein synthesis. So what happens in this case is that this EIF2 alpha, uh, it will convert this, it will binds with the GDP, that is guanine diphosphate, and helps converting this GDP into the GTP, that is guanine diphosphate, into the guanine triphosphate. And this guanine triphosphate, when it is bind with alpha, beta and gamma of EIF2 alpha, uh, then it will interact with methionine tRNA. And we know from one of our previous videos that this methionine is complementary to the start codon, that is AUG, that can be found in the mRNA. And when this methionine at this step 2, the methionine tRNA will bind to the EIF2 alpha subunit. This whole thing is together called as EIF2 alpha subunit. And when it is bind to methionine tRNA, then it is called as EIF2 GTP methionine tRNA, or ternary complex. And this ternary complex will attract the small subunit of ribosome, that is 40S subunit. And along with that, the other it will also attract other initiation factor, and together it will form a 43S pre-initiation complex. When this ribosome subunit and a other initiation factor binds with this ternary complex. After that, this pre-initiation complex will interact with the cap, or 5 prime end of the mRNA, and starts reading the mRNA till it finds the starts codon, that is AUG. And when it finds the AUG, what will happen is it will hydrolyze this GTP, that is shown in yellow color. It will hydrolyze the GTP to GDP, and as soon as it hydrolyzes, it will released from this structure, and this ternary tRNA methionine will bind to the AUG, and with the release of this structure, that is EIF2 alpha, it helps, or it creates the space for 60S ribosome subunit to bind to this complex. And when this both 60S subunit and 40S subunit binds along with the initiation factors, then it is together complex, and it is together called as ATS initiation complex. And this will further continue the translation process of mRNA till it reaches the poly A chain, or we can say that till it reaches the stop codon. This is how the normal mRNA process works. However, in case of phosphorylation of this EIF2 alpha, what will happen is that it will not uh, convert this GDP into the GTP, and as this form is uh, less available in case of phosphorylation of EIF2 alpha, then this will affect the initiation of the mRNA translation. And overall impact, we can consider it in a way that it will reduce the mRNA translation whenever this EIF2 alpha is phosphorylated. So the this is how the phosphorylation of this complex, that is EIF2, will affect the mRNA translation process globally. So let us understand what are some of the conditions at which mRNA can be affected. So first we will consider the normal condition, that is physiological condition. In this case, this ternary complex is not limited, and since this ternary complex, the initial form of ternary complex is not phosphorylated, so this ternary complex is not limited. So when the a ribosome starts reading upstream ORF1, then it will starts recruiting the ternary complex, and by the time it starts to read this upstream ORF2 mRNA sequence, then it will have enough ternary complex to translate this open reading frame 2. So when the turns come for the translation of main open reading frame, this translation of upstream open reading frame 2 will not be able to, will prevent the translation of main open reading frame in case of normal condition. However, in case of stressed condition, and we have discussed some of the stress condition in our previous videos, we can see that there is epigenetic changes occurs in case of stress conditions such as in ER stress. Why this happens? Because in case of stress condition, this ternary complex is limited, and one of the reason is that the EIF2 alpha could be phosphorylated, and because of this, while during the translation of this upstream open reading frame 1, it will not have, it will not starts to recruit, or it will not recruit at same capacity as in case of a normal condition, this ternary complex. And so because of this, the translation of upstream open reading frame 2 will not be completed by the time main open reading frame translation starts. And when this when the translation of main open reading frame starts, it will leads to epigenetic changes that might that is essential at that point of time in order to reverse back the cell from the stress condition to the normal condition. So this is how this mRNA translation process occurs normally, and how this epigenetic changes occurs in case of stress condition. I hope you have understood or got some of the basic idea. In order to understand this thing in details, you can go through the description. The link has been provided for the paper from which these figures has been taken. So you can understand this thing in details from the original paper that has been published. So that's all for this video. Thank you for your time.